Hey, ever tried Japanese sake? I'm from Japan and today I will make a simple version and explain the science behind it. Let's get started. Start by putting freshly cooked rice and water into a clean jar and mix well. Next, add the koji and yeast and then mix again. Cover the jar with plastic wrap and loose a lid and let it sit in the cool, dark place. 3 days later, did it actually become sake? いただきます。おお、酒だ。But how can this happen just by mixing the ingredients? Let's take a step back. See the bubbles in this time lapse? That's science at work. The stars here are koji and yeast named Saccharomyces cerevisiae, performing multiple parallel fermentation. Koji enzymes break down rice starch into glucose that yeast can use. S. cerevisiae converts glucose through glycolysis to form pyruvate, generating NADH and ATP. Instead of entering the citric acid cycle, most pyruvate is converted to acetaldehyde and then to ethanol by alcohol dehydrogenase. The bubble you saw comes from this carbon dioxide. Why not use a more efficient citric acid cycle? Due to the Crabtree effect. Yeast produce ethanol even in the presence of oxygen when sugar is abundant, inhibiting competitors and give ethanol-tolerant strains an advantage. By the way, you might notice something unusual in the recipe. We are using baker yeast. Why could it make sake? The answer is that both baker's yeast and sake yeast descend from a common ancestor. Also, while the strain likely originated in East Asia, it's still unclear how this yeast entered human communities and became established, termed primary domestication. Two hypotheses are presented. The single domestication hypothesis posits that domesticated strains originated from a single domestication event of a common wild ancestor, while the multiple domestication hypothesis suggests that multiple lineages of wild strain were independently domesticated. The single domestication hypothesis is supported by the low genetic diversity, suggesting a possible bottleneck event. Conversely, the multiple domestication hypothesis is supported by the fact that different domesticated lineages each become similar to different wild lineages. These discrepancies likely result from limited sample data. Regardless of the hypothesis, yeast later became specialized for human use through repeated selection, a process called secondary domestication. Let's examine each of the adapted genetic traits. For example, in wine, strains have increased copper resistance genes due to copper sulfide pesticide, and have also evolved improved sugar utilization efficiency. Next, in some bread strains, higher copy number of genes involved in methanol production, imparting a toasty aroma and richness. In cheese, S. cerevisiae may have acquired genes from other species through horizontal gene transfer, HGT, allowing it to rapidly use galactose left over from lactose breakdown and better adapt to the fermentation environment. In environment created by humans, yeast populations are densely packed and subject to intense competition for nutrients, leading to frequent mating as a response to stress. Furthermore, selection pressure are less stringent than in nature, making mutations more likely to accumulate. These characteristics serve as evidence that yeast has evolved to adapt the human environment. Then, how does sake yeast change their genes? Sake yeast was shown to originate from koji rice in the late 1800s, and a superior strain were collected and distributed. The representative strain, Kyokai No. 7, produced more ethanol than the other S. cerevisiae strains due to a loss of function mutation in Lim15. Later, in 1976, Kyokai No. 11 was isolated from K7. It showed enhanced alcohol tolerance due to the activation of stress response genes such as HSP12. In this way, the tasty foods and drinks we enjoy come from the amazing adaptation of S. cerevisiae.